Welcome student on another video lecture. Topic is opium. Today we are going to discuss about biogenesis of morphine. Here there are structural relationship between benzene isoquinoline alkaloid and dihydroxyphenylalanine, and these are used for derivation of morphine, and during that it will occur due to rotation of tetrahydroisoquinoline residue, and which is followed by oxidative ring closure. So there will be the rotation of tetrahydroisoquinoline residue, and which is followed by oxidative ring closure. Oxidative ring closure means fused 5 and 6 membered ring without ring junction of heteroatom, that is known as oxidative ring closure. In an experiment, labeled tyrosine is administered to poppy capsule, and here two precursors are demonstrated, were incorporated into morphine molecule. The intermediate stage was confirmed by demonstration of norlaudanozolene, which act as a more efficient precursor for morphine than tyrosine. So when these two precursors are compared, that is norlaudanozolene and tyrosine, the norlaudanozolene is more efficient precursor for incorporation into morphine molecule than tyrosine. By cultivation of poppy plant in 14 CO2 and by injection of label alkaloid into the plant, it was shown that the first major alkaloid form is thebane, and which is irreversibly converted to codeine and then to morphine. So when the cultivation of poppy plant is taken out in presence of 14 CO2, and the plants are injected with label alkaloidal compound, and its biosynthetic pathway is demonstrated, or then it will found that the major alkaloid first form is thebane, and that thebane is then converted into codeine and then to morphine. So here norlaudanozolene, it is converted into laudanosine by using an enzyme SAM. It belongs to a superfamily enzyme, and which is a metal containing enzyme, and that is used for conversion of norlaudanozolene to laudanosine. That laudanosine is converted into papaverine by using an enzyme PAPAD, which is a flavin adenine dinucleotide enzyme, and which is a redox active coenzyme associated with various protein, and it is also used in various enzymatic reactions of metabolism. So norlaudanozolene is converted into laudanosine, which is converted into papaverine. When this norlaudanozolene is incorporated into papaverine and thebane, it is at higher rate than tyrosine, so it is more efficient precursor as compared to tyrosine. After that, norlaudanozolene is converted into laudanosine. Laudanosolene is converted to reticulin. So norlaudanozolene is converted into laudanosolene, and that will be converted into reticulin. Reticulin, these are the enantiomers. S and R enantiomers. S is showing anti-clockwise direction, whereas R is in clockwise direction. So these are the two enantiomers which are further used for formation of morphine. So reticulin, that is converted to 1,2-dihydroreticulinium ion by using an enzyme reticulin oxidase. Reticulin oxidase enzyme is used, and that enzyme is a oxidoreductase enzyme, which specifically acting on XH and YH to form a XY bond with oxygen as acceptor, and so it is a oxidoreductase enzyme that will convert into ion. Reticulinium ions are produced here, ionic form you can see here. Now NADPH dependent reductase enzyme that will convert that reticulinium ion to reticulin R. Here the S form is converted into R form, in S configuration is converted into R configuration, and by using an enzyme NADPH dependent reductase. This enzyme is involved in detoxification of reactive carbonyls in plant. Whatever the reactive carbonyls they are present, they can be detoxified by using this NADPH dependent reductase, and now the S form is converted into R form. Now R reticulin, that will be converted to salutaridine by using an enzyme heme dependent salutaridine synthase. Heme dependent salitaridine synthase is the enzyme which is used or which catalyzes the formation of morphine in alkaloid salitaridine by intermolecular phenol oxidation. So intermolecular phenolic oxidation will occur in R reticulin. In this R reticulin intermolecular phenolic oxidation will occur, but incorporation of without incorporation of oxygen into the product. The oxygen will not incorporate into the product, but phenolic oxidation, you can see here, phenolic oxidation will occur and it will convert it into salitaridine. So due to phenolic oxidation, reticulin is converted into salitaridine. Now that salitaridine is converted to salitaridinol by using an enzyme salitaridine reductase. So salitaridine reductase enzyme is used for conversion of salitaridine to salitaridinol, and salitaridine reductase enzyme which involved in biosynthesis of morphine by benzene isoquinoline alkaloid, and which catalyzes specifically stereospecific conversion of salitaridine to salitaridinol. That salitaridinol is converted into salitaridinol 7-O acetate by using an enzyme salitaridinol acyl transferase. So this enzyme which catalyzes the chemical reaction of acetyl coenzyme A and salitaridinol. So here the chemical reaction will occur between acetyl coenzyme A and salitaridinol, and it will produce a salitaridinol 7-O acetate. So here combination of acetyl coenzyme plus salitaridinol will takes place and it will form a salitaridinol 7-O acetate. Also here acetylation will occur, acetyl group is attached here and it will produce a salitaridinol 7-O acetate. Now that salitaridinol 7-O acetate spontaneously converted to thebane. So here the first alkaloid which is gets synthesized, that is thebane. It is a major alkaloid which is formed, that is thebane. Now that thebane undergo demethylation. Thebane will undergo demethylation. This methyl group will remove and it will produce a neopenone, and that will convert it to codenone. Here from thebane now codenone is produced. Now that codenone is converted to codeine by using an enzyme codenone reductase that will reduce the codenone to codeine. So that reductase enzyme will reduce codenone to codeine, and due to demethylation of that codeine, now the morphine is produced. So here finally you are getting a morphine here. From reticulin to salitaridinol 7-O acetate, thebane, codeine, and then morphine is produced. In this way, three alkaloids, that is thebane, codeine, and morphine, they will get synthesized by this biosynthetic pathway. So if you like my video, please press the like button and subscribe my channel. Thank you.